Hello students, good morning. Yesterday we finished structure of testis, histological study of testis. In that we have seen different parts, okay, which type of cells are involved in spermatogenesis, overall process of spermatogenesis, not in detail, that we are going to study later on, okay. Today especially I prepared one presentation because I wanted to show you some diagrams from NCERT, because all diagrams are very important and questions can come on those diagrams, okay. So for that sake I made a very simple presentation so that you can also understand what type of questions can ask from the diagram, okay. So the first diagram which I have taken, that is of male reproductive system. Same diagram it is given in your NCERT, okay. If you can see, already we have studied the structure of testes. Now that we have revised so many times, so I am not going to explain that part again, okay. So pair of testes in scrotal sac, from there start the vas deferens. Now just see here, vas deferens, when it comes near to the urinary bladder, its ends swell, okay. That swollen portion is known as ampulla. That labeling is not given in the diagram, okay. So if you want, you can label that part. So vas deferens, when it comes near the opening of urethra, okay, it is swollen, that is known as ampulla. Fine. Now in case of human male, urethra, it is a common passage for sperms as well as for urine, okay. So secretions of sperms as well as secretion of glands, okay, all they are carried by the common passage, that is urethra, okay. So this vas deferens, okay, it enters into urethra. Now already we have learned that in seminiferous tubules, sperms are produced through different duct system. They are stored in epididymis, where the sperms attains functional maturity, and once they mature, then they are carried by this vas deferens, okay. Now from there into the urethra. Now after that there are different glands, accessory glands, okay. From that first pair, slightly leaf-like larger gland, that is known as seminal vesicle, okay. Below that there is a prostate gland, then small knob-like bulbourethral gland. These glands secretes their secretions also into the urethra, and then the semen, that part we will see later on, and then it is carried or it is ejaculated through the external genitalia, that is the penis, okay. Now this diagram from NCERT, this diagram comes instead of this name, they put here A, B, C, D, and they ask us to identify or what is the path of sperm from the seminiferous tubule to means up to ejaculation, what is the path of sperms, fine. So that sequence also you have to remember. So the study for board point of view also, this diagram comes to draw. So just do practice for drawing also this diagram, okay. This diagram can be asked for drawing, okay. So first diagram of male reproductive system, okay, understood. Now let's move. Yesterday we studied in detail the histology of seminiferous tubule. So specially I made one slide here so that you can understand the internal structure. As we studied before, seminiferous tubules have two types of cells: triangular, slightly conical, larger cells, okay. These are known as Sertoli cells, okay, and these are somatic cells. Then second group of cells, these are your germ cells, okay, germinal cuboidal epithelium. These are known as spermatogonia. When you study the transverse section of seminiferous tubule, you observe this spermatogonia at different stages of development. As we discussed yesterday, okay, these spermatogonia, they differentiate into primary spermatocytes. These primary spermatocytes undergo meiotic division to produce secondary spermatocytes, fine. Secondary spermatocyte complete second meiotic division, become spermatids. Yes, these spermatids mature, they obtain their nutrition, by embedding into membrane of Sertoli cell, okay, mature and then become spermatozoa or sperms. Fine. Now, throughout their development, these spermatozoa or sperm, they are embedded in the membrane of Sertoli cell. We discussed functions performed by Sertoli cells also. Quickly we will revise that part. First one, it provides nutrition to the developing sperms so that we can call them as a nurse cell. They sustain overall development of spermatogenesis. So Sertoli cells are also called as sustentacular cells. So Sertoli cells or nurse cells or sustentacular cells. Fine. Same name. Okay. Now they phagocyte the dead and damaged sperms. Okay. Then they secrete anti-Mullerian factor. So that point I discussed yesterday, the Sertoli cell secrete anti-Mullerian factor. Now Mullerian duct in the embryonic cell develop into oviduct. So to prevent the formation of oviduct, okay, and uterus, anti-Mullerian factor is secreted. Then Sertoli cell form the blood barrier system. Now we discussed what is the blood barrier system. Spermatogonia never come in direct contact with the blood. So whatever materials to be transferred, these materials are transferred through Sertoli cell. Fine. These are functions performed by Sertoli cell. Outside or in between the seminiferous tubule, the fluid, it is known as interstitial fluid. These interstitial fluids have aggregates of endocrine cells. These are known as Leydig cells or interstitial cells. These cells secretes androgens. Androgens, it is a group of male hormones. Okay, like testosterone, dihydrotestosterone. Okay, so all these hormones we will learn afterwards. So this is the seminiferous tubule. Questions will come on this part. Okay. Then let's move ahead. Fine. Till now we completed the primary sex organ, that is testis. Now let's move towards secondary sex organs. Now secondary sex organs means glands, ducts, okay, and external genitalia. From these ducts already we have learned when we studied the structure of testis. How these ducts, they help in the transfer of gametes. Next important part that we are going to study, that is the accessory glands. Okay, in the first diagram we studied, if you have NCERT textbook with you, just keep that diagram in front of you so that you can understand where are they located. Okay, now there are three types of glands: seminal vesicle, prostate gland, and bulbourethral or Cowper's gland. Right? Now let's see the first one, seminal vesicles. Seminal vesicles are paired glands, okay, tubular and coiled. If you have diagram in front of you, just simultaneously just observe that diagram. Okay, these are paired glands, tubular, coiled. They are larger in size. They are located behind the bladder. Okay, its position is important. They are located behind the bladder. Also called as uterus masculinus. Okay, very important point. Just make a star for this or just underline the statement. Seminal vesicles are also called as uterus masculinus. Okay. Now seminal vesicles are formed from Mullerian duct of embryo. Just now we discussed that Sertoli cells secrete anti-Mullerian factor to prevent formation of oviduct, or to prevent formation of female reproductive structures and to promote development of male reproductive structures. So these seminal vesicles, they form from Mullerian duct of embryo. Seminal vesicles are internally lined by glandular epithelium. Again, important point, just underline it, lined by glandular epithelium. Now, seminal vesicles secrete a fluid that is known as seminal fluid. Fine. This is viscous, lubricating, transparent, jelly-like fluid. Okay. It forms about 60 to 70% part of semen. Okay. It is slightly alkaline with pH of 7.3. Now, its components are very important. Okay, because these can asked in exam. So seminal fluid or the seminal plasma, it is also known as seminal plasma, contains fructose, citric acid, prostaglandins, inositol and some proteins. Fine. Important is fructose because as you know that sperms travel up to egg. Sperms have to carry or sperms have to do a very long journey. Okay, they have to reach up to the egg cell for fertilization. For their transfer, lot of energy is required. So to provide energy in the motility of sperm, fructose it is present. So that fructose provide the instant energy. Okay, when sperms are transferred, when they reach or when they are swimming to reach up to the egg cell. So this fructose is present in seminal vesicle. It is secreted by seminal vesicle, present in the seminal fluid. Along with fructose, there is citric acid, prostaglandins, inositol as well as proteins. So the first gland, seminal vesicle. Okay. Now let's see the second one. The second gland, which is unpaired single gland, it is prostate gland. If you see its shape, it is slightly chestnut shape, okay, and present at the base of bladder. It surrounds the first part of urethra, upper part of urethra. Okay, it is collection of 30 to 40 tubulo-alveolar glands. Where you have learned tubulo-alveolar gland, just remember animal tissues. Okay, types of glands. Fine. Tubulo-alveolar glands. So about 30 to 40 tubulo-alveolar glands, they unite, okay, and they form a single prostate gland. It is unpaired, but in man it is about 5-lobe structure. Okay. It also secretes the alkaline fluid that is known as prostatic fluid. It is also milky, sticky, thick, slightly jelly-like, and it forms about 30% of semen. Now this fluid helps in activation of sperms. It provides characteristic odor to the seminal fluid. Okay. Then it also secretes citric acid, calcium, potassium, some clotting enzyme, profibrinolysin, okay, etc. Now see here this point, it is very important. What is this clotting or what is the role of these clotting enzymes and profibrinolysin? Okay, now see, secretions of seminal vesicles and prostate gland, okay, help in the coagulation of semen or the sperm. See, semen means what? Semen, it is the secretion or it is aggregation of sperms, okay, and secretion of these glands, okay, that forms the semen. At the time of sexual intercourse, semen is or sperms are ejaculated, okay, along with the secretions of these glands. Okay, so secretion of seminal vesicle and prostate gland, they help in the coagulation of semen. So it is deposited in the female vaginal tract in a coagulated form. Okay, where mobility of sperms, it is reduced. But as soon as sperms reach or after ejaculation, due to this fibrinolysins, semen again liquefies and sperm can move. So these profibrinolysis, okay, these are the inactive form. As soon as they come in contact with the acidic pH of vagina, they become active fibrinolysins, okay. The coagulated semen get normal, sperms become active, and now they can migrate toward egg cell. Fine. I hope you understood this point. I will explain this part again. At the time of ejaculation, semen is coagulated. It is slightly in concentrated form, okay, so that it can be deposited in the female vaginal tract properly. So for that place, secretions of seminal vesicle and prostate glands are helping for that. Now, after ejaculation, the coagulated sperms have to get the normal speed, okay, or they have to be active. So in that case, profibrinolysis, which are present in the prostatic fluid, okay, become active. Okay, profibrinolysin, it is an inactive form. When it comes in contact with the acidic pH of vagina, in males, the secretions of prostate gland, seminal vesicle, they are alkaline, okay, whereas vagina has an acidic pH, okay, or the secretions of vagina, they have acidic pH. So whenever profibrinolysis, they come in contact with the vaginal pH, become active, and coagulated semen become, these your sperms become active, and then they move toward egg cell for fertilization. Again, semen liquefies, sperm become active, okay, and sperms can move to fertilize the egg cell. Fine. Now, prostatitis, it is the inflammation of prostate gland. Now, one more thing, in India, cancer of prostate gland, it is more common in male. As you know, in case of females, breast cancer is more common, like that. Okay, in males, prostate gland cancer, it is more common. Right? So this is about prostate gland. Okay. Now let's see the third one. The third and the last paired gland is called as Cowper's glands or bulbourethral gland. Okay, these are paired, small-sized, pea-shaped or pea-sized. Okay, located on the lateral sides of urethra. Okay, at the base of penis. You have diagram in front of you. You can see the location. It also secretes transparent, slimy, jelly-like fluid, which is slightly alkaline. It has also pH 7.2. It also helps in reducing the acidity of urethra, cleans it for the movement of sperm. Next point we will discuss, that is semen. What is semen? Okay, semen, it is nothing but, as I told you, sperm plus secretions of glands. It is known as semen. As we discussed now, seminal vesicle secretes seminal fluid or seminal plasma. Prostate gland also secrete prostatic fluid, right? Cowper's gland also secrete fluid. All these fluids, they are alkaline in nature. Why they are alkaline in nature? Because vaginal pH, it is acidic. So to neutralize that pH, it is highly acidic. Okay. So in that acidic pH, most of the sperms may get killed. So to neutralize the acidic pH of vagina, okay, the pH of secretions of these glands, it is alkaline. Okay, the normal volume or amount of semen, it is about 2.5 to 5 ml per ejaculation. Okay, slightly alkaline because secretions of all these glands, they are alkaline. So obviously semen is also slightly alkaline. It has pH of 7.2 to 7.4. Right? It provides nutrition as well as transportation medium to sperm. So we discussed this point before. Sperm transfer through this medium only. The normal sperm count is about 200 to 300 million sperms. Okay, this is the norm, or about 20 to 20 million per ml. Now from this, for normal fertility, 60% must have normal shape and size, and at least 40% must show vigorous motility. On these statements question comes, sometimes they ask some numericals also on this point. Okay, so for normal fertility, out of these many sperms, millions of sperms, 60% must have normal shape and size, and from that at least 40% must show vigorous motility. Okay, now there is one clinical condition that is known as oligospermia. In case of oligospermia, if the sperm count is less than 20 million per ml, it is known as oligospermia. It may lead to sterility. Okay, and one more clinical condition known as azoospermia. Okay, in azoospermia, either there is absence or near absence of sperm, so obviously it will also lead to the sterility. Right? So this is the composition of semen. Next part we will see, that is the structure of urethra. Okay, as we discussed, urethra, it is a common passage for urine as well as for semen. Okay, so it provides common pathway for the flow of urine and semen. It is much longer in males than in females. It is about 20 centimeters long, and based on its position, urethra has three parts. One simple diagram I have given for your reference. If you want, you can draw it, otherwise just see it for study. It has three parts. First one, it is known as prostatic or glandular urethra. Obviously from name you can understand the part of urethra which is surrounded by a prostate gland, okay, which is upper vein, so it is short, proximal and surrounded by a prostate gland. So that part of urethra, it is known as prostatic or glandular urethra, okay. The second one, that is membranous urethra. It is very short, present just below the prostatic urethra. It is middle and without any type of covering. And the next one, that is the penile urethra. It is long, distal and passes through the penis. So the part of urethra which is present throughout the penis, okay, it is known as penile urethra, okay. So three parts of urethra: prostatic or glandular urethra, membranous urethra and penile urethra, okay. Okay, one more point I have added over here regarding sphincters present in urethra. So two important sphincters are present. One, that is the internal sphincter of smooth muscle. It is located at the beginning of urethra. And the another, that is external sphincter of striated muscles located around the membranous part. So only you have to remember where are they located. Okay? And the last secondary sex organ, this is the ejaculatory structure, okay, or you can say copulatory organ, external genitalia, okay, that is the penis. Now external genitalia, those organs, those parts which help in deposition of male gametes, they are known as external genitalia, okay. So in males, external genitalia, it is penis, okay. So penis, it is a muscular and tubular organ about 15 centimeter long, okay. It is erectile copulatory intromittent organ. It helps in the deposition of sperms, okay. Its terminal part is bulging and it is called as glans penis and covered by a loose folds of movable skin, that is known as foreskin or prepuce. If you have diagram in front of you, just see that diagram, okay. The terminal part of penis, it is bulging, slightly swollen. It is known as glans penis. It is covered by a loose movable skin or folds of skin, that is known as foreskin or prepuce, okay. These foreskin or prepuce, they have special glands. These glands are known as prepucial glands or Tyson's gland, okay. They secrete a kind of sebaceous substance that is known as smegma, okay. They secrete a kind of sebaceous substance that is known as smegma. There is one clinical condition where prepuce is removed by surgery, that is known as circumcision. Why it is removed? Because of smegma, bacterial infection can occur in foreskin or prepuce. So it can lead to the inflammation. So to prevent that infection or inflammation, that prepuce or foreskin, it is removed by surgery, that is known as circumcision, okay. Now this is just introductory information or some information about penis. So here I have shown the internal structure of penis, okay. So if you see the internal structure of penis, first point we can easily find that your penile urethra, okay, it passes through the penis. So that is also shown here, penile urethra, okay. Interiorly, penis, it is formed by three cylindrical cords of spongy and erectile tissues, okay. Internally, or the interior of penis, it is formed by three cylindrical cord or masses of spongy and erectile tissues. Two of these tissues or cords, they are thicker and situated parallelly on right and left side, okay. It forms the thick part of penis that remains in front of penis, okay. Now these two cords are known as corpora cavernosa, okay. Here you can find two larger masses of those erectile and spongy tissue. They are known as corpora cavernosa, okay. The fibers of tunica albuginea surrounds both the cords, okay. jointly and also form a separate sheath surrounding to each cord. Okay, that is not shown over here, but outside to each unit, outside to each corpora cavernosa, there is a layer that is known as tunica albuginea. Okay, this tunica albuginea is present surrounding to each bundle as well as surrounding to both the bundles also. Okay, that is tunica albugina. Some fibers form a partition that is called as septum penis between these cords. Okay, between these cords some fibers can also run, that is known as septum penis. Okay, the third and the smaller cord or group of tissue forms the part of penis which remain inferior anterior in the erect penis. Urethra runs through this cord, and now that is known as corpus urethrae or corpus spongiosum. So the single unpaired smaller mass of tissue through this penile urethra runs, it is known as corpus urethrae or corpus spongiosum. Fine, you understood there are three types of erectile tissues. Okay, two are present in group that is known as corpora cavernosa. They are larger in size. The one which is smaller through which the penile urethra runs, okay, it is known as corpus or the corpora spongiosum, okay, or corpus urethrae. Fine, understood, simple. Rarely questions will come, only questions can be asked related to corpora cavernosa, corpora spongiosum, through which part penile urethra runs. Okay, along with this there are some blood vessels also deep dorsal, because for erection of penis, okay, blood supply is required, so blood vessels also move through it. Okay, so in the transverse section we can also observe that artery, dorsal artery, deep dorsal vein. But the thing which you have to remember in this structure, that is two bundles of corpora cavernosa and one bundle of corpus spongiosum. Fine, this point you have to remember. And the last point in this, that is erection of penis. Okay, how or what causes the erection of penis? Now, as it is supplied with blood vessels, so it is vascular. It is controlled by autonomous nervous system. Okay, so at the time of ejaculation, blood supply is increased, and that causes the dilation of penile arteries. Okay, because of this there is an enlargement and hardening of penis. Okay, this helps in the deposition of semen or for the ejaculation. Okay, so these are the different parts of male reproductive system. I hope you understood all the parts. Just go through this video again. Just note down this point. Okay, most of the things which we learned today, these things are not mentioned in the NCERT, but these are very important to understand. So with this we finished the first part, that is parts of male or the components of male reproductive system. Okay, and the next part that we will learn tomorrow, that is hormonal regulation of male reproductive system along with spermatogenesis. Very important point. Okay, every time questions will come from that part. So tomorrow we will study hormonal regulation of male reproductive system and spermatogenesis. I hope you all are revising this lesson, not only these lessons but previous lessons also. You are solving sheets which you have. If you are solving, then no one is sending any doubts, okay, based on this, because anytime your test can be arranged. Okay, so for that sake you have to do the regular studies. Okay, so this we completed the first part, that is components of male reproductive system. Fine students, tomorrow again we will meet, okay, and we will start hormonal regulation and spermatogenesis in males. After that we will start to female reproductive system.